Namaste students, Vidyapeeth. I, JV and Dr. Chandrakala, Assistant Professor, Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the reproduction in gymnosperms. As we have already discussed about the gymnosperms, its characteristic distribution, habitat, and morphological and anatomical structures. So, in today's session, we are going to discuss about the reproduction and reproduction. Here, the vegetative methods of reproduction are very rare in gymnosperms, but cyclids do propagate through bulbils. Sexual reproduction, gymnosperms are heterosporous, producing two kinds of spores in different spore-bearing structures, that is megasporangium or ovule, or the microsporangium or the polar sac. Okay? So, the gymnosperm may be monoecious in Pinus and Cedar, or dioecious in Cypress and Aphridia, Ginkgo, and the male bearing and the female Ginkgo and the bear male and female ovum, except in Cypress female is a female plant. The size of the male ovum ranges from 1 mm like in Jamia and 60 centimeter in copper. The position of the strobili on the plant from the terminal to the lateral, or in cycles, the microsporangiophils are loosely arranged between the successive cones of the foliage leaves. There are various variations in the arrangement of the female cones or the, we can say, strobili, which you can like the ovules are generally borne on the adaxial or the upper surface of the megasporophils or the ovuferous scales. The ovule is generally orthotropous. The spore mother cells contained in the sporangia undergo meiosis to form respectively the microspores and the megaspores that represent the first cell of the haploid or the gametophytic generation. The microsporangium contains numerous microspores where the megasporangium contains only one megaspore. Okay? So the gametophytes are endosporic. So endosporic means they develop within the respective spore walls. Okay? So the microspore forms the microgametophyte, or we can say it as a pollen grain that produce the male gametes, or we can say it as a sperm. The megaspore developed into the megasporophyte or megagametophytes. We can call it as also female gametophyte that bears archegonium containing the female gamete, that is the egg. So the microsporangia and the male gametophytes. Microsporangia are borne on the adaxial surface of the microspore. The development of the microsporangium is as follows. The initial form of the archeosporangial cells that lead to the formation of the wall layer and the sporogenesis cells. The later give rise to the microspore mother cells, which divide mitotically to form the tetrad of the microspores. The microspores of the first cells of the male gametophytes, and these usually undergo the divisions of form one or more root hallus cells and the enthelial initium. The development of the male gametophyte being endospermic or it takes part in partly in the microsporangium or partly in the pollen chamber of the ovule. The microspores are like central cells or the prothali. The central cell is again like divided into antheridial initial or prothali cells of antheridial initial uh, undergo like antheridial cells to tube cells. So the various variations, the number of prothali cells or the increase in the motility of the male gametes and then time to formation and discharge. At the time of shedding of the pollen grains are usually multicelled and contain one or two or more prothali cells. The tube of the nucleus and the antheria cells. The pollen grains are winged in pinus or non-winged in cyclus or Jaipur. Either the male gametophyte lacks prothali or the number of fringes from the cyclus. The ovules or the female gametophytes. The ovules are known as the naked it's or bone on the adaxial surface or the metasporangial cells, which in turn are generally arranged spirally around the central bone. The ovules are sessile and are orthotrophous and each contain of the parent gamete's mass of the cells or nucleus or the microsporangium. The nucleus become enveloped by the single massive integument and grow around its leaves. A small opening of the micropyles at the tip and the megasporangium enclosed by the integument of and is called the ovule. The nucleus contains a small diploid. The megasporangium cells undergo meiosis to form the tetrad of the diploid microscope, of which only one remains and the function and other three degenerate. The functional microspore which is usually in the gelagil end of the microspores enlarges and undergoes pre-nuclear division. Thus, the young gametophyte develops within the megaspore. The centripetal walls of forms or the starts around the pre-nuclei of the female gametophytes. The integument of the gametophyte or like here the integument in the gymnosperms ovules consist of three layers, which we can study in our later chapters. We will study in detail regarding this. Here the archaeoniums are large and long and lacks the neck canal cells. Natum lacks distinct archegonium, egg, nuclei, and prothalus tubule containing the nucleus, egg, nuclei, respectively. Thank you. In our next session, we will be going to discuss about the pollination and fertilization. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, please write down in the comment box. We will resolve it in the problem solving